Namaskar, my dear students. Hope you are all fine. Today I am going to talk about the second part of the chapter Plant Growth and Development. And the topic, after discussing about the definition of the uh, growth, development, various zones of maturation, zone of elongation, zone of cell growth, differentiation, de-differentiation, redifferentiation, plasticity. Now I am going to talk about the plant growth regulators today. As you know, the plant growth regulators are the small, simple molecules of the diverse chemical composition, and they are of various types. In fact, these are the chemical signals. The plant growth regulators are the chemical signals that controls and coordinate growth, metabolism, and morphogenesis of the plant. Plant hormones are called as the plant growth regulating substances because most of the plant hormones, they are related with one or the other aspect of the growth. In fact, these are the organic compound molecules made in one part of the plant, and their response, either it is produced at the point where they are formed, or they are transported to other part where it elicits a response. They are made in several locations in the plant body, and they produce their effect in different places or at the same place. Broadly, the plant growth hormones, they are the plant growth regulators, commonly called as PGRs, plant growth regulators. They are broadly classified into two groups based on their functions in the plant body. Firstly, one group of PGRs are involved in growth promoting activities such as cell division, cell enlargement, tropic growth, flowering, fruiting, seed formation, etc. These are called as the plant growth hormones, and these are auxins, gibberellins, and cytokinins. Auxin, gibberellins, and cytokinins. These are called as the plant growth regulators. The PGRs of the other group play an important role in plant responses to wounds, stresses of biotic and abiotic origin. They are also involved in various growth inhibiting activities such as dormancy, senescence, etc. And these includes the ethylene and the abscisic acid. And they fit in the second group and are largely called as the inhibitor of the growth activities. Whatsoever is their nature of function, whether they are the growth promoters or the inhibitors, ultimately they are regulating the growth, and the five groups under which we discuss them, they are the auxins, gibberellins, cytokinins, ethylene, and abscisic acid. We shall be discussing these growth regulators one by one, and to start with, I will talk about the auxins, one of the very widely discussed and having a wide effect on the growth of the plants. The group is the auxins, the first growth regulators. Let us talk first about the discovery of the auxins. Here I must mention that it is very interesting that most of the plant growth regulators, they were accidentally discovered. However, discovery of the auxins, this was first group of the plant hormones discovered in 1881 by Charles and Francis Darwin. They reported experiments on response of growing plants to light associated with the phototropic response. In fact, uh, they were working on the canary grass, which responded to the unilateral illumination by growing towards the light source, that is phototropism. After a series of experiments, it was concluded that tip of the coleoptile was the site of the transmittable influence that caused the bending of the entire coleoptile, and auxins were isolated by FW vent from the tips of the coleoptile of the oats seedling. However, they occur in very low concentrations and were first isolated from human urine and in coleoptiles of the oats seedlings. They show different response to the growth patterns if they are provided in different doses, and let us discuss their various uh, natures on what are the. First of all, let us discuss about the types of the auxins. I must mention here that there are the auxins which occur naturally in the plant, and nowadays some auxins are synthetically made in order to offer them for the growth of the plant. Now the natural auxins, these are naturally occurring in plants and therefore they are regarded as the phytohormones. The best known natural auxin is indole 3 acetic acid, and to the other natural and synthetic compounds having certain growth regulating properties. Now the synthetic auxins, these are many chemicals synthesized in laboratories that cause various physiological responses similar to indole 3 acetic acid. Synthetic auxins are not considered as phytohormones, and most of them are the indole butyric acid IBA that has been isolated from plants, then naphthalene acetic acid, then 2,4-D, 2,4-dichlorophenoxyacetic acid. These are the synthetic auxins. All these auxins have been extensively used in agricultural and the horticultural practices. Now, let us discuss what are the various functions of the auxins or what are the various physiological effects of auxins. Auxin being a diverse hormone or the growth regulator, depending upon the time at which it is monitored to the plant or on the dose in which it is present, it shows various physiological effects, and these effects are cell elongation, apical dominance, root initiation, prevention of abscission, parthenocarpy, respiration, callus formation, and vascular differentiation. I will discuss these in little detail. The auxins help to initiate rooting in the stem cutting, that is root initiation, rooting in stem cutting, and application widely used for plant propagation. Auxins, they promote flowering. For example, in pineapple, they help to prevent fruit and leaf drop at the early stages but promote the abscission of the older mature leaves and the fruits. In most higher plants, the growing apical bud inhibits the growth of the lateral auxiliary bud, and that is called as the apical dominance. What is apical dominance? The apical bud, it grows and inhibits the growth of the lateral buds. The removal of the shoot tips, that is called as the decapitation, usually results in the growth of the lateral buds. It is widely applied in tea plantations, hedge making, because the auxins, when they are produced on the tips of the plant, and if the tip is removed, it will initiate the lateral growth. Auxins also induce parthenocarpy. For example, in tomatoes, they are widely used as herbicides. 2,4-D, that is 2,4-dichlorophenoxyacetic acid, is widely used to kill dicotyledonous weed because they do not affect the mature monocotyledonous plants. It is used to prepare weed-free lawns by gardeners. Auxins also control xylem differentiation and therefore it helps in the cell division. So these are the various physiological effects of auxins, and now we shall be discussing the second hormone, and that is the gibberellins. So far as discovery of the gibberellins is concerned, Prior to World War II, Japanese scientists were investigating a rice disease that made the plant uh, very tall, but it prevented the grain formation. They showed that a chemical was responsible and they caused it a fungal growth that caused the hyperelongation. And in Japanese language, they called it bakene, which means foolish seedling, disease of the rice seedling. This was caused by a fungal pathogen, Gibberella fujikori. E. Kurosawa reported the appearance of symptom. Kurosawa, he reported the symptoms of the disease in an uninfected rice seedling when they were treated with sterile filtrate of the fungus. The active substance was later identified as gibberellic acid. In the 1915, this chemical was isolated in US and UK and was called as the gibberellic acid. F. Skoog and his co-worker observed that from the internodal segments of the tobacco stems, a callus, what is callus? You know, mass of undifferentiated cell in which no morphogenesis has occurred, that proliferated only if in addition to auxins, the nutrient medium was supplemented with one of the following that extract, that is coconut milk, DNA, etc., which was later identified, crystallized, and was found that it was gibberellic acid. So far as the gibberellins are concerned, many gibberellins differing in their biological activities, they have been discovered and their number is now over 100. That is a very important another kind of the promotory plant growth regulator, which are now 100 in their number, and these gibberellins, they have been reported from widely different organisms such as fungi and higher plants. These are denoted as GA1, that is gibberellic acid 1, gibberellic acid 2, gibberellic acid 3, GA2, GA3, and so on. However, gibberellic acid 3, that is GA3, was one of the first gibberellins to be discovered and remains most extensively studied of all the other forms. Now, how does and where does its biosynthesis of the gibberellins in the plants occurs? Biosynthesized in apical tissues, and there are three main sites of their biosynthesis. And what are these three main sites? First is the developing fruits and seeds, young leaves of developing apical buds, and apical regions of the roots. These are the three regions where the biosynthesis of the gibberellins takes place in the higher plants. Now, pathway of gibberellin biosynthesis can be divided into two stages, each of which is accomplished in a different cellular compartments that I shall be discussing like this when I discuss the mechanism of the action. It acts by inducing activity of gluconeogenic enzyme during early stages of the seed germination. Gibberellins also induce a synthesis of alpha amylase and other hydrolytic enzyme during germination of monocot seeds. Gibberellins also mobilizes seed storage reserves during seed germination and seed emergence. They produce a wide range of physiological responses in the plants. Gibberellins have the ability to cause increase in length of the axis, and that is used to increase the length of grape stalks. Gibberellins cause fruits like apple to elongate and improve its shape. They also delay senescence. Thus, fruits can be left on the tree longer so as to extend their market period. GA3 or gibberellic acid 3 is used to speed up the malting process in brewing industry. Spraying juvenile conifers with gibberellic acid hastens the maturity period, thus leading to early seed production. Gibberellins also promote bolting. Bolting means increased internodal elongation just prior to flowering in beet, cabbage and many plants with rosette habit. So gibberellin is the hormone responsible for the increase in the length at various stages in various ways. So after gibberellins, we shall be discussing the third hormone, and that is cytokinins. Cytokinins have specific effect, and let us first discuss the discovery of the cytokinins. Cytokinins are the plant growth hormones which are basic in nature, either immunopurines or phenylurea derivatives that promote cytokinesis. Cytokinesis, you have studied in the process of cell division, mitosis and meiosis. Cytokinesis means cell division and subsequently formation of the cell wall or the cell membrane. Either it works alone or in combination with the auxins and the other hormones. Now, F. Skoog and his co-workers observed that from the internodal stem segments of the tobacco stem, the callus was formed. As I discussed with you earlier, they found that in addition to auxins, the nutrient medium, when was supplemented with the coconut milk or DNA, they found that it was containing cytokinins, which promotes active substance termed as kinetin. And these proved that there is a substance which is responsible for the cell division to occur. They also analyzed to find out the growth promoting chemical, and first cytokinin was discovered from the degraded from autoclaved herring sperm DNA by Miller in the year 1955. Now, where does they occur and how does they occur? The cytokinins, they are found in the cells of the plants, and they occur in the free form or in the conjugated form. tRNA cyto are formed in every living cell in the cytoplasm, chloroplast and mitochondria. So, they occur free or in the conjugated form and are found in the cell cytoplasm, chloroplast and mitochondria. Now let us discuss the types of the cytokinins, and broadly we have classified cytokinins into two types: adenine type cytokinin, which include kinetin, zeatin and 6-benzylaminoprines, and they are mostly synthesized in roots. The adenine type cytokinins, they are mostly synthesized in roots. There is another group of cytokinins which are phenylurea type cytokinin, for example TDZ, diphenylurea and thidiazuron, and these are found not found in plants, but they are artificially present or are obtained from some other sources. Now what does the cytokinins do? Cytokinins are specific effect, that is cytokinins, and since the discovery of the cytokinins, it has been found that it produces the new cells, and therefore it promotes cell division. Because it produces new cells, it helps in the morphogenesis. It helps in the formation of new leaves, chloroplast in the leaves. It lateral shoot growth and adventitious shoots are formed. So it helps in the lateral bud development, that suppressing the effect of the apical dominance. Since it helps in the cell division, therefore it delays the process of senescence. That is, the senescence is delayed when cytokinins are applied to the plants. They overcome the effect of apical dominance. They promote the nutrient mobilization in the different parts of the plant, and mobilization of the nutrients helps in delaying the senescence. They also play a role in stomatal opening and rapid transport in the xylem stream. The rapid transport in xylem stream delays the senescence and their effect on the lateral buds that causes the uh, effect of the apical dominance to be overcome. So these are the various functions of the cytokinins. Now we shall be discussing a very important hormone, the only gaseous hormone, and that is ethylene. Now what is ethylene doing in the plants? Let us first discuss its history. Increase in the ethylene production was associated with peak in respiration during tomato ripening. Therefore, ethylene is also called as the fruit ripening agent when applied in very small amount. This endogenous growth regulator as a plant hormone in an amount that can reach a very slow, very lesser amount, it causes fruit ripening, because once it is given in larger amounts, it shows some other effects which may not be good for the plant, and it shows many other functions. Let us discuss these functions. Ethylene is gaseous in form and hence shows the rapid diffusion. It affects the adjacent individuals. What is that? Uh, if Because it is gaseous hormone, it travels from one fruit to the other and hence hastens the process of fruit ripening. It causes senescence and abscission. It interferes with the auxin transport. It inhibits the stem elongation. Its positive feedback mechanism amplify responses in organism. Amplification occurs when the stimulus is further activated, which initiates an additional response that produces system change. I will explain my point that influences of ethylene on the plant includes horizontal growth of the seedling, swelling of the axis, and apical hook formation in dicot seedling. It promotes senescence and abscission of the plant organ, especially of the leaves and flowers. Ethylene is highly effective in fruit ripening, enhances the respiration rate, and right in the respiration rate is called respiratory climactic. The respiratory climactic is the term which is means the rate of the respiration is enhanced in response to the ethylene hormones. Ethylene breaks down the seed and the bud dormancy, initiates germination in peanut seeds, sprouting of the potato tubers. Ethylene promotes rapid internode petiole elongation in deep water rice plants. Ethylene is used to initiate flowering and for synchronizing fruit set in pineapple. The most widely used compound as a source of ethylene is ethyphone. Now, ethyphone hastens fruit ripening in tomato and apples, accelerates abscission in flowers and fruits of some plants like coconut, cherry, walnut. It promotes female flowering in cucumber, thereby increasing its yield. That is what I mean to say by the amplifying effects of the cytokinins. So these are the various functions, not cytokinin, functions of the ethylene. Now, next we will be discussing the abscisic acid. Abscisic acid is also called as the stress hormone. Abscisic acid is an inhibitory hormone in plants that help in the plant adapt to the stress. It was discovered for its role in regulating abscission and dormancy. But like other plant growth regulator, it has also showed wide-ranging effects on the plant growth and development. It acts as a general plant growth inhibitor and inhibitor of plant metabolism. It allows the plant to survive during the times of adversity, but also assists the plant in adapting to the changing seasons. So Abscisic acid plays a number of roles, and abscisic acid is a single compound unlike auxins, gibberellins and cytokinins, where the number of compounds are categorized under gibberellins, auxins and cytokinin. It was abscission 2 originally, because it was thought to play a major role in abscission of the plant, also called as the stress hormone. The abscisic acid stimulates the closure of stomata in epidermis and increases the tolerance in the plants to various kinds of stresses. That's why called as the stress hormone. Now, what is abscission? Let me explain to you, which is considered to be the major role of the abscisic acid. Abscisic acid promotes abscission of these fruits and the leaves, in contrast to auxin, which inhibits abscission. It is in fact this action that gave rise to the name of the plant, abscisic acid. Abscission means breaking down of the plant organ by deadening of its tissue. Abscisic acid acts as an antagonistic to growth regulators or the gibberellins. That so we can summarize that for every phase of the growth, differentiation and the fruit, one or the other plant growth regulator has some role to play. Such roles could be complementary or antagonistic. They could be individualistic or synergistic. So after the discussing these growth hormones, we shall describe the role of light and temperature. As of course, light and temperature are extrinsic factors and growth regulators are the intrinsic factors, sometimes applied from the outside. We shall be discussing these two things, and first is the photoperiodism, that is the effect of the light on the plant, and then we will be discussing vernalization. Let's first talk about photoperiodism. The plants, it has been observed that some plants require a periodic exposure to light to induce flowering. It has been seen that such plants are able to measure the duration of the exposure of the light. The plants, in order to flower, require certain day length, that is the relative length of the day and the night which the plant requires for the flowering to occur is called its photoperiod. It can be defined as a response of the plant to photoperiod expressed in the form of flowering, and that phenomena is called as photoperiodism. Now, photoperiodism, it is the duration of the light to which the plant shows response for flowering to take place. Every plant requires a definite length of the day. Day means light. The former, the group of, we have grouped the plants according to their duration of the light which they need for the flowering to occur, and the three groups are long day plants, short day plants and the day neutral plants. For example, some plants require the exposure of the light for a period succeeding even more than the well-defined critical duration, while others will be exposed to light for a period lesser than the critical duration before the flowering is initiated in them. The group where they require more hours of the light than the usual hours, they are called as the long day plant. They require more than 12 hours of light for the flowering to occur. In the same way, the critical duration of the light is suppose 12 hours, and if a plant requires only lesser than the 12 hours or the more hours of darkness, that is here you can see 18 hours of darkness, they are called as the short day plants. It would not be wrong to call it long night plant instead of short day plant. The critical duration is different for different plants. There are many plants, however, where there is no such correlation between exposure of light and induction of flowering response. Such plants are called as the day neutral plants. it is now known that the duration of the light period is not, but the duration of the dark period is also of the equal importance. Hence it is said that the flowering in such plants, certain plant, does not depend on the combination of the day and dark exposure, but also on their relative duration. The response of the plant to periods of day as well as night, it causes a photoperiodism. And it is also interesting to note that while shoot apices modify themselves into flowering apices prior to flowering, they, that is shoot apices of the plants by themselves cannot perceive photoperiod. The site of perception of the light and darkness are the leaves, because it has been hypothesized that there is a hormone substance that is responsible for the flowering, and that hormone is called as the phytochrome, which actually exists in two versions. One is PR, that absorbs red light, and PF4, that absorbs the far red light, and it occurs in two variants, that is PF4 for red light and PR, that is the red light. This hormonal substance is produced in the leaves in response to the duration of the night or the day, and it migrates from the leaves to the shoot apices for inducing flowering only when the plants are exposed to necessary inductive photoperiod, and these two forms are interconvertible, PR and PFR. Now, after the photoperiodism, we will be discussing another extrinsic factor, and that is the effect of temperature, to which we call vernalization. Let us discuss it. What is vernalization? There are the plants for which the flowering is either quantitatively or qualitatively depending on the exposure of the low temperature. This phenomena is called as vernalization. So we may say that vernalization is the process in which low temperature given as a result shows the makes the plant to show flowering, or it is the process whereby the flowering is promoted by cold temperature treatment given to a fully hydrated seed or to a growing plant. That is, low temperature treatment to the plant in order to flower is called as vernalization. Now, vernalization, how does it occur? That is the mechanism of vernalization. The stimulus received by the actively dividing cells of the shoot or the embryo tip travels to all parts of the plant and prepare it to flower. The stimulus that is considered to be a hormone-like substance and has been called as the vernalin. Of course, the vernalin is produced inside the plant, but its stimulus is the low temperature, which is extrinsic factor. It can be passed from one plant to the other through grafting in case of henbane, but not seen in many plants. It prevents precocious reproductive development late in the flower growing season and enables the plant to have sufficient time to reach maturity. Now, uh, vernalization refers specifically to the promotion of the flowering by period of low temperature treatment. Some important food plants such as wheat, barley, rye have two kinds of varieties, winter variety and spring varieties. The spring varieties are normally planted in spring and come to flower and produce grains before the end of the growing season. Winter varieties, however, planted in spring would normally fail to flower in the flowering season. They only germinate after they have overcome the winter, because only the low temperature is the stimulus by which they start the flowering. Other examples of vernalization is seen in biennial plants, such as monocarpic plants. Sugar beet, cabbage, carrots are some of the common biennial plants which require the cold temperature treatment, which stimulates a subsequent photoperiodic flowering response. Now these are the factors which affect the flowering. First is the water and flowering due to vernalization. First is the water and oxygen. Seeds need to imbibe 10 to 20% water. Dry seeds cannot be vernalized. So not mere the cold temperature treatments, but the other factors must be minimal in order of the flowering to be produced. Only then the cold temperature treatment or the vernalization would work. Similarly, oxygen is used during respiration. The energy is provided by the respiration. So inhibition of respiration blocks the process of vernalization. Temperature, of course, this is a temperature dependent process. The seeds are exposed to low temperature during vernalization. Temperature, if lower than 4 degrees centigrade, is not effective. Similarly, temperature above 12 degrees centigrade does not cause vernalization. The duration of the vernalization, however, varies in different species. Sometimes a devernalization is done. What is that? Exposing of the vernalized seeds to high temperature, they lose the effect of vernalization, and this phenomena is called as the devernalization, which may be needed in some cases in order to increase the length of the uh, duration, so that the late flowering can be induced if it is of use to the mankind. So this is all about the growth regulators and the growth factors and the extrinsic factors, the light and the temperature, which shows differential effects on the growth and development of the plants. So that's all for this chapter. Thank you so much, my dear students. So I would suggest you to go through the NCERT books and also go through this lecture so as to understand the lecture fully, and that's all for this chapter. Thank you.